Good morning, Ilya.
Good morning, Nick. I don't see. Oh, okay. So you're already ahead of me, huh? How did you do this? Share, huh? When I hit share, do you see this? Nick. Do you see my presentation?
Yeah, I can see your presentation.
You see this?
Yeah.
Because when you go to a full screen presentation, all right, kill this.
It's on presenter view though.
So you see the current slide and the next, right?
Yeah, right. And I can see your notes if you have any.
Okay, I'll change this. Are
you working with one screen or two screens?
I'm working with two screens, and I have a third one.
Oh, geez. So let me show you mine real quick.
Okay, go ahead.
Can you just see my presentation and not my notes?
No notes.
Right, just the presentation.
Yes. How do I do this?
On the one screen, I have like all my slides, and then on the other screen is my presenter view. I'm just sharing one screen.
Okay, so I will share. Okay. So this is, you see now the presentation, right?
Correct.
Yeah, but you also see the slides, the list of the slides, which will not. What about now?
I just see the slides, but I still see like the outline, you know.
You see the outline, or you don't see the outline?
Yeah, I can see. Yeah, I still see the outline of stuff.
How do I hide? Huh?
What's that?
How do I hide the outline that anybody or nobody can see the outline?
I think it's the way your PowerPoint set up. You want the presenter view to be in two windows. Or on two screens, I'm sorry.
Slideshow, always use subtitles. No. Okay, so now if I hit, you see the slide, the next slide, and the outline.
Correct, I see presenter view.
We don't like this.
No.
So now you see the presentation only with or without the outline.
With the outline.
How do I get rid of this?
Let me Google it.
Okay, now you see everything, right? Is Tony there? No, no Tony. Okay.
Господин Левтеров, имате ли ме?
Стоян Йовчев is talking?
Да.
Окей. Добре, значи минаваме за бързо на български. Проблема е, че аз се свързвам с Mac и цялата информация на цялата презентация се вижда като аутлайн отляво. Имаш ли някаква представа как мога да затрия аз това нещо? Да се вижда само самата презентация.
Аз малко мак не разбирам, обаче видях, че ви е в слайдшоу режим. Тоест разделен на два екрана. Това ли е проблема?
Е?
Това, че се разделя на два екрана ли е проблема?
Okay, I have a problem with the sound as well.
You need to click on the slideshow tab.
Yes, and I am in the slideshow.
No, yeah, the top slideshow tab.
Най-отгоре
And then-
има.
Click on monitor.
There is no monitor here. It's a Mac.
Oh,
Play from the current slide, okay, but that is not what we want. No.
Той иска да си изключите слайдшоу режима, това, че ви разделя на два екрана. Горе има един тап, на който пише слайдшоу. Някъде 6 или 7 подред.
То е така.
Кликнете го.
То е така. Обаче, когато кликна там, и аз познавам много добре този етап, това, което виждам аз е
Presenter view.
From current slide, play from start, presenter view.
Това Presenter view. Трябва да се изключи и когато минете в слайдшоу режим, няма да е на два екрана, ще бъде само на един.
Presenter.
Да.
Presenter view.
Да. Това трябва да бъде изключено.
Окей, сега какво виждаш?
Това не беше настройката за активиране и спиране, направо го пуска. Може ли да отидем пак там?
Да, разбира се. Slideshow.
Ако... без пикване... Може да правим втората опция. Там, където бяхте току-що.
Така, значи първата опция е play from start.
Да, не, преди малко бяхте на някакви настройки.
Да. Ето ги настройките.
Да. На първото show type, може ли да изберем второто и да опитаме отново?
Да.
Да видим какво ще стане. И сега на slideshow. Сега виждаме един екран.
Сега виждаш един екран.
Да, голям.
Един голям екран. Добре.
Не виждам следващия слайд в момента.
Не го виждаш, но го виждаш сега.
Да, сега го виждам, само по един ми показва.
Okay, so the problem is that I don't see anybody except myself. But that's okay.
You can see me, for example.
How do we stop the spelling and the recording? Okay. Do you really want to see the talk where I'm talking or not? It's going to be irritating because you will always be able to see this. I believe it's an option from your side. You have to fill this.
Mm-hmm.
All right. I believe you don't see the subtitles now, do you?
Oh, looks good.
Huh?
I said, no, it looks good.
Hey. Is the organizer, I see him. The problem is that I see only one person, the most important person. This is Professor Township, I believe.
Hey, how are you guys?
We're doing okay.
Ready to start?
I believe so.
Yeah, we have a number of people already attending, so I think we...
Not so many people?
No, I think more people will join in anytime. So.
It's up to you, Tony. We have time. It's up to you. Tell me.
Let's start. So I'd like to greet Dr. Receptor and Dr. Fitz in today's second part session of our online workshop. After discussing the physical and biophysical features of cell site separation and isolation, today we go at the subcellular level, discussing one particular organelle, the extracellular vesicles, microsomes, as biomarkers in human disease. Both of our lecturers are from the University of Pittsburgh, and both of them are working in the field of neurodegeneration and Alzheimer's disease, so field of neuroscience. And this is what they are going to mostly show, but many of the things they're going to show us actually refer to, I think, more broadly to the use of extracellular vesicles as biomarkers. And I think that is why this topic is interesting. In the future, if possible, we are also going to organize some meeting on the use of extracellular vesicles in cancer, perhaps. But with the national program of European research networks, we are focused on the field of neuroscience, and that is why our lecturers will focus on the use of extracellular vesicles in the field of neuroscience in particular. So I would like to again acknowledge the generous contribution of the Bulgarian Ministry of Education and Science, which made this whole workshop starting Friday and continuing today possible. On the website you have seen, those of you who don't know Ilya and Nicholas, we have seen their CVs. We have published briefly. Ilya is more than 20 years now working in the field of Alzheimer's disease research. Together with Dr. Koldamova, they lead a group at the University of Pittsburgh, utilizing animal models as well as human samples to study different aspects of the disease and to try to generate ideas for treatment and diagnosis. So the extracellular vesicles are a very small aspect of their work, very small part of their work, but it's a relatively recent, exciting development in many fields, including in this field. And that is why we are organizing this online course, because there are people also in our university who are interested in that. And as we are going to be, this is also streamed on the YouTube, so students can watch it. And we are very grateful to you, Ilya and Nicholas, that you will share your experience on this topic. So we are looking forward to start. What we discussed previously was that the two presentations will follow back to back, and then we make a discussion, tips and tricks, and this kind of questions and answers. Is that so? You said that you would prefer to go like this instead of having questions after each of the talks.
Okay.
Yeah, I think
What do you
that's-
think?
Yeah, that's a good format.
Okay, so maybe then Dr. Lefterov, you can start.
Okay. Good morning, everybody. Thank you very much, Tony, on behalf of myself and Nick. We appreciate very much the opportunity to share with you what we are doing, not to the full extent, but the most important part of our work, particularly in the field of extracellular vesicles as a diagnostic markers in Alzheimer's disease. I have to admit that in 40-45 minutes it is almost impossible to present in a full extent, or even in a very concise form, what is the situation with extracellular vesicles, because this is extremely fast developing field in any field of human pathology and physiology. To make it easier on each of the slides, and people who are, and I see people very experienced in general in functional biochemistry. Diana Ivanova is there, I see. So I believe many of you... What happened? Okay. Hi, Diana. What I'm saying is that many of you, I believe, are well aware of the pathways and biochemical processes that I'm going to talk about. But to make it easier on each of the slides, I tried because there are too many abbreviations, too many symbols, too many parts of particular biochemical pathways that are abbreviated. I tried to explain the abbreviations on the slides. And if you have any questions at the end of the two presentations, we will be happy to answer those. So, as you have already heard, the first talk will be biogenesis and function of extracellular vesicles. And I can tell you before I start that on each of the slides, you will have so much information that each of the slides by itself can be a separate presentation, but that's the nature of the research in the field of extracellular vesicles. So I will try at the same time to present the function of extracellular vesicles in a way that you probably haven't seen or heard so far, because we have been struggling with this, I would say, several years now, and I don't believe that extracellular vesicles are mostly vehicles to transfer and to transpose regulatory molecules from one cell to another cell. Extracellular vesicles by nature have a different role. Their physiological role is slightly different, and you will see that it is kind of difficult. Even now, it is almost 40 years since the discovery of or the description of extracellular vesicles, and we don't have a good idea how their cargo works as regulatory tools in neighboring or remote cells. So the question here is, as I understand this, do they remotely supply regulatory weapons, or they are just public servants as diagnostic tools and biomarkers? So if we put the title in this way, we have to... I'm sorry? Can I continue? Tony? Tony?
Sure, you can.
I can, because I hear voices from here and there. Okay, so the definition of weapons, and I want you to be careful here. Maybe the weapons may be construed to include anything used to gain a tactical, strategic, material, or mental advantage over an adversary or enemy target. So this is the definition of weapons, and you will see that the function of extracellular vesicles mostly falls into this definition. So the discovery of extracellular vesicles and early research, one of the first, probably not the very first papers that described extracellular vesicles was published in 1983, and this is a milestone paper which describes the extracellular vesicles and their release as part of a disposal mechanism to discard unwanted materials from cells. And I will show you that this is a definition of extracellular vesicles and their function that we can consider established, very well established. So for more than, for almost 25 years since the first paper, in 2007, Vladi et al., the senior author of this Nature Cell Biology paper is now editor-in-chief of a Journal of Extracellular Vesicles. So they demonstrated that exosome-mediated transfer of mRNAs and microRNAs is a novel mechanism of genetic exchange between cells. Yes, it is an exchange. So microRNAs and messenger RNAs that were detected and identified in extracellular vesicles could be the same, identified and named or positioned in a genome in a neighboring cell. So that's fine. And then in 2012 and then in 2013, another two milestone papers have been published by Fruweis and his colleagues that demonstrated the release of extracellular vesicles is an important mediator of intercellular communication involved in normal physiological process as well as in pathological progression. In other words, by the beginning of, or let's say 5 years, more than 5 years back from now, we knew about extracellular vesicles as vehicles for intercellular communication and subcellular structures with very, very important cargo. So now, this is an important point in understanding of the biogenesis of extracellular vesicles. And I'm absolutely sure that most of you know very well the pathway of receptor-mediated endocytosis. So receptor-mediated endocytosis is part of endosomal pathway. An endosomal pathway includes pinocytosis, phagocytosis, receptor-mediated ligand complex, and they all converge into receptor-mediated endocytosis-derived early endosomes. These early endosomes form multivesicular bodies, and by membrane fusion, they either go to lysosomes, or they include subcellular structures from the cytoplasm, a process called macroautophagy, part of microautophagy structures. And the role of all these, as you can see, is just the neutralization and degradation of bad stuff, quote unquote, that enters the cells. So this is endosomal pathway in grosso modo. There are many, many, many directions that follow from within the endosomal pathway, but that's not our goal to talk about them now. So this is a summary of the endosomal pathway and autophagy. So, but we will introduce here the exosome generation or biogenesis and secretion. And you will see that these two processes, exosome generation, biogenesis, and secretion and autophagy, are very, very tightly connected. So here is the endosomal pathway: early endosome, late endosome, multivesicular body. We mentioned microautophagy, macroautophagy, and then endosomal pathway is actually a recycling mechanism, but part of these multivesicular bodies can serve as vehicles for secretion of exosomes. So we're not going to talk about phagophore. We're not talking about ubiquitinated proteins, RNA-induced silencing complexes, microRNAs, aggregated harmful proteins, misfolded proteins. All of these have one very specific goal: to get rid of unwanted material that entered the cell, or unwanted proteins, misfolded and aggregated proteins that should be exported outside the cells. So this is one very complicated mechanism, and somewhere here we see that exosomes can be released. Now, the formation of intraluminal vesicles within multivesicular bodies is the start of the biogenesis of exosomes. So, as I said, here are the abbreviations of the many proteins that you will see here. What we have to remember from this slide is that there are two pathways for biogenesis, or generation of exosomes. One is X-cord endosomal sorting complexes required for transport dependent, X-cord dependent pathway, and the other is X-cord independent pathway. Each of these have multiple, 3 or 4 so-called sub-pathways, because there are multiple X-cord complexes, 0, 1, 2, 3, and each of them forms complexes, higher order of complexes with different proteins, in order to internalize multivesicular bodies. So the protein composition of these microvesicular bodies, multivesicular bodies, is slightly different. For example, TSG101 protein is more in X-cord independent generated multivesicular bodies, but not something that is missing in this X-cord dependent biogenesis encounter. So once again, two methods for biogenesis, or two pathways for biogenesis of multivesicular bodies that will serve as an initial vehicle for release of exosomes: X-cord dependent and X-cord independent. Now, how these extracellular vesicles are released? There are two major pathways for release of extracellular vesicles. The first one is fusion of multivesicular bodies with the outer layer of the plasma membrane, resulting in the release of these exosomes. And you can see these RAP proteins. I don't know, maybe some of you have been or are working on RAP proteins. See, this is number 11, number 35, number 27. Altogether, they are 70. And they are members of RAS superfamily of small G proteins. And you see other proteins here, Helix, lump. Maybe you will notice acetylcholine esterase, which is a protein involved in pathogenesis of Alzheimer's disease. People thought, some of them still think. You can just imagine how complicated this process, this pathways is. And the other one is a pathway or machinery called outward budding. infusion of the plasma membrane controlled by different proteins in extracellular signaling. So these are different, but I would say slightly different, because you see, we talked about TSG101 is here, TSG101 is here, and you see, Arab proteins are also here. The question is if there are preferential conditions for this or this machinery to get assembled and release the extracellular vesicles. The common understanding now is that this outward budding of multivesicular bodies occurs in conditions of cellular stress. I always get headache when I hear cellular stress and stress-activated mechanisms, but this is what the people think is the preferential condition for the activation of outward budding. Now, this is I would say, a summary of the extracellular vesicles generation, biogenesis, and signaling between neighboring cells. So until 2018, until early 2019, people were talking, and a lot of papers have been published. A lot of papers are being published now with exosomes in the titles and in the abstracts and everywhere in the papers. Now, Nick will talk about this in more details, but since 2018, the guidelines from the International Society for Extracellular Vesicles is very clear. Now we talk about extracellular vesicles of different sizes. So we have already talked about the generation and the release of extracellular vesicles. So extracellular vesicles of different sizes have the same biogenesis, but they may have different functions and communication mechanisms once they are outside of the cells. Now, the smallest are exomers, which are of the size 35 nanometers and below. Their exact mechanism of release is unknown. The next are extracellular vesicles, which originate in early endosomal pathway. We talked about this. Early endosomal pathway ends up with the generation of multivesicular bodies, and they are included in microvesicular bodies. Fuse with plasma membrane and then secrete. They fuse with plasma membrane and then they secrete extracellular vesicles. These extracellular vesicles that are secreted by fusion of multivesicular bodies can be small, medium, and apoptotic bodies. Small, between 40 and 200 nanometers. Medium, between 200 nanometers and 1 micrometer. And apoptotic bodies are big. So, as you can see, this is the problem. Medium to large and small extracellular vesicles overlap. I will show you this later. And this is important because there is nothing that we can use right now to differentiate these two classes of extracellular vesicles. Now, this is the cargo of extracellular vesicles. As you can see, the cargo of extracellular vesicles is actually everything that you know as big molecules and molecular complexes. So we have DNA, we have microRNAs, we have non-coding RNAs, we have all kinds, not only heat shock proteins, we have enzymes, we have messenger RNA, and membrane transport and fusion proteins, all the jungle of biochemistry. So very, very rich structures. How do we address the questions what these members of the cargo or what the cargo by itself, by large, is doing? A very, very difficult question. I personally don't think that we have a clear answer even how to address the question of function of each of these members of the cargo of extracellular vesicles. Now, here is an example. We are, as Tony said, we are very much interested in communication, intercellular communications between major brain cell types. And you know that excluding cells in the vascular structures in brain, the major brain cell types are oligodendrocytes, neurons, microglia, and astrocytes. Now here we have glioblastoma cells because glioblastoma is one of the loveliest. And I apologize for the term, but one of the liveliest type of brain cancer that people working with extracellular vesicles use as structures to learn more about extracellular vesicles. So not only glioblastoma, all kind of brain tumors release extracellular vesicles. Extracellular vesicles are released by oligodendrocytes. They are released by neurons, and they enter the brain through blood-brain barrier. They are circulatory extracellular vesicles, and we know not everything, but we know a lot about circulatory extracellular vesicles, and I can tell you that they definitely enter the brain not only through the blood-brain barrier, but also through blood-CSF barrier. Remember the choroid plexus and the epithelial cell layer and ependymal cell layer, so they enter the brain. Now, what is happening is that all these extracellular vesicles go towards microglia. And we can imagine that most of those are internalized through endosomal pathway, and their destiny is destruction, degradation of the stuff that they carry, their cargo. Now, there are, of course, exceptions. I'm not saying that this is the only function of the microcellular extracellular vesicles. So you see that here they can transfer information which is critical for neuronal pruning, synaptic pruning, and also some people think that extracellular vesicles secreted from neurons and loaded with A beta or tau go to microglia, and of course the easiest explanation is that they go there to be destroyed. So they go there for degradation of A beta and tau. One thing that we know for sure is that microglia is one of the main pathways for degradation of amyloid beta and tau. So here is a very raw picture of what happens in brain and how extracellular vesicles travel and traffic in brain. So, because I'm going to talk mostly about RNA cargo in exosomes, the first thing that I want to show you is that there are many types of RNA molecules in the exosomes. Messenger RNAs, microRNAs, transfer RNAs, ribosomal RNAs, long non-coding RNAs, small non-coding RNAs, yRNAs, PIWI RNAs, and circular RNAs. Now, this is something that inspired researchers from Sweden and Finland in 2013 to suggest that because of this cargo, extracellular vesicles, once released, can go through interstitial space or through circulation to neighboring or remote cells and exert a regulatory role, because these RNAs, some of them, not all of them, have proven regulatory roles. Now, if it is true, yes, it is true. To what extent, we don't know. So, how does it work in the real life? I will briefly tell you about the current research that we have in our laboratory, and I apologize that I cannot show you everything because there are publication and some patent issues, and we cannot ignore those. But whatever is possible, I will show you here. And afterwards, you can ask and we can talk without a record if you want to learn more. So the goal of our research now is to investigate extracellular vesicles in plasma of Alzheimer's disease patients and controls, and to look for members of the cargo that we can use to discriminate Alzheimer's disease patients and control individuals. Control individuals mean non-demented individuals. So these are kind of not well separated populations, but there are clinical indications where and how to discriminate these cohorts. So the first thing is to characterize by size and concentration extracellular vesicles. So as you can see, the peak here, these are the extracellular vesicles that we isolate. These are the extracellular vesicle size of the markers, of the of the markers that we use. So as you can see, yes, it is within very close. The peak is very close to 100 nanometers, but there are plenty, not very many, but enough extracellular vesicles with the borderline size that falls between small and medium size extracellular vesicles. So here are the protein markers that we use to prove that we have enrichment of proteins that are markers for extracellular vesicles. Now, this guy, TSG 101. So the fact that we have some preservation of TSG101 tells you that we most probably have extracellular vesicles that are escort dependent generated. But this is just a hypothesis or suggestion, because this TSG101 may come from extracellular vesicles generated by escort independent mechanisms. So this is the first step of your research if you want to characterize and investigate extracellular vesicles. Now, I told you that we are very much interested in non-coding RNAs, particularly small non-coding RNAs. So once you have the extracellular vesicles characterized, you isolate RNA, and this RNA, because you use a particular type of kits, the RNAs are highly enriched in small size RNAs. Most of them are non-coding, or all of them are non-coding. So then you have to sequence these RNAs. Nowadays, it is very easy, because if you have the RNA isolated, you don't even have to generate libraries. You just send the RNAs to a sequencing core or company. They will generate the libraries and they will sequence. And now it is good, because within less than 3 weeks or 2 weeks, you will have the result. The result is your FASTA-Q files, the sequencing files. Now, just a few words about a huge consortium funded by NIH maybe 4 years ago. And these guys did an outstanding job. So they generated a pipeline called Exert, and the pipeline is available on Jamboree web server. So you have your sequencing files, and you can upload. Of course, there is an account, but everything is free, funded by NIH. You upload your sequencing file to Jamboree web server. Now, you can, if you have a computational power, you can install the Jamboree or the Exert pipeline on your local machines. The problem here is that sometimes, not all the times, but sometimes the processing of the sequencing files is very challenging, power demanding, computational power demanding, and it may take more time than you get done on Jamboree web server. But anyway, so you can do this outside your laboratory. So then once you have your Jamboree or your Exert pipeline output, these are the steps that you go through very easy. It is possible to learn how to work with Exert pipeline in less than an hour. If you have your FASTA-Q files sequenced, if you have your FASTA-Q files processed by the web server. So pre-processing, filtering, then at any of these steps, you actually get rid of unmapped reads. And by unmapped reads, it means reads that fall within non-coding small RNAs. So there is a endogenous alignment, depending on your source, human or mouse, endogenous filtering, and then exogenous alignment, and you get rid of that as well, because you don't want exogenous RNAs, and you know that in extracellular vesicles they can be found. So then the user can prioritize the order in which annotations, Myrna base, tRNA scan, pRNA, gen code, circular RNA, used for quantification based on confidence in the presence of a given RNA biotype in a given sample. Let me tell you what it means. You are interested only in Myrnas, in microRNAs. There is a step that you get rid of all the other non-coding small RNAs, and you concentrate on your microRNAs, and vice versa. Let's say that you are interested in small nucleolar RNAs. Then you use gen code to scan for SNORNs and small nuclear RNAs, and you detect the newest examples of those non-coding RNAs. The most recent database, including all known at the moment, small nucleolar RNAs and small nuclear RNAs, is SNO database. You can, if you are interested, we will provide all the websites, all the addresses, so you can try this. No matter what kind of sequencing files you have, you can try this pipeline. So once you have the sequencing done, this is the distribution of non-coding RNAs that you may get. I can promise you that you will see differences in these percentages, but they are almost, or they are similar in different samples that you use for isolating RNAs, extracellular vesicles, or maybe not extracellular vesicles, because small non-coding RNAs are found in protein RNA complexes and lipoprotein RNA complexes, and Nick will talk about this. So see, the biggest chunk in your non-coding non-coding RNAs are microRNAs, followed by transfer exogenous RNAs. You cannot get rid of those, even though they are pretty big and effort put on the generation of well-working filters. piRNAs, miscellaneous RNAs, protein-coding RNAs. These are parts of protein-coding RNAs. These are actually parts of my mRNAs, messenger RNAs, mitochondrial ribosomal RNAs, mitochondrial transfer, small nucleolar RNAs of CD box. We will talk about this briefly, and other RNAs. So 5 more minutes and I'm done probably. Type and size distribution of human RNAs. It is very, very helpful diagram, because you see that they are small, they are mid-size, and they are long. You have to learn very well the functional, the biogenesis of these small RNAs if you are concentrated on microRNAs or on piRNAs. TRFs are transfer RNA-derived fragments. So in this field, we have small Cajal RNAs, we have small nucleolar RNAs, transfer RNAs, and all kinds of other RNAs. You are not interested in long human RNAs unless your goal is long non-coding RNAs. So here is the field that most of the non-coding RNAs of huge interest now are positioned. Now, I guess you are well aware of biogenesis of microRNAs and how do they work. I put this slide here just to emphasize that the biogenesis of any type of non-coding RNAs is very similar, if not the same, except, of course, different protein complexes, protein RNA complexes, and enzymes are involved. So step number one, transcription, then processing, intranuclear processing, then export, then processing in the cytoplasm, association with proteins, formation of complexes, nuclear protein complexes, and then translational repression, mRNA deadenylation, and mRNA cleavage. These are the mechanisms of action of microRNAs. So the steps, you remember steps. So this is small nucleolar RNAs. And how do they work? Transcription, some processing in the nucleus, no export through the nuclear membrane, but then once they are assembled with proteins and cleaved at both flanks, they are transferred to the nucleolus. So these are mature SNOR nuclear proteins. Once again, transcription, processing, transfer through a membrane. So small nucleolar RNAs are very interesting non-coding RNAs because they work as guides for big RNA protein complexes that have two major, and for now, two only known functions to modify RNAs. So SNORNA guides the modifications of a big machinery, and depending on the type of the small nucleolar RNAs, the two modifications are methylation or pseudo-uridylation. This is important because changing the methyl sites and the ribosyl sites changes the function or the efficiency of the function of mostly transfer RNAs. Now, Pi RNA biogenesis. We can spend a lot of time, but again, one thing that is that prompted people to call and to question if endosmall inhibitory RNA exists, if endosmall RNA inhibitors exist as a precursor, is the biogenesis of piRNAs. Again, transcription, piRNA precursors are here. They go outside the cells, the nucleus, in the cytoplasm. They form complexes with proteins. Then they enter the so-called ping pong circle, form another more complicated protein complexes, and go back into the nucleus. So what they do in the nucleus is that they mediate transcriptional silencing, and which is called endogenous transcriptional silencing. So these huge protein complexes are engaged in the ping pong cycle, and they actually methylate transcription sites or transcription or promoters or sites immediately preceding transposons. And in this case, they play a big role in silencing transposons in the specific genomic sites. And we know this for mice. Oops, not available. We know this happens in mouse cells. We don't know how efficient is this process in humans, but I can tell you that we published recently, 2 years ago, a paper in PLOS genetics showing that piRNAs play very important role in testicular tissues of rats, and they have a very interesting regulatory role depending on the alcohol consumption in male rats. So, biogenesis and function of extracellular vesicles. I don't know if I convinced you that they are weapons, or they are public servants as diagnostic tools and biomarkers. Nick will talk about those options and possibilities right now. These are the people who are part of the team Extracellular Vesicles in Alzheimer's disease. This is just half of the laboratory. Myself, Rada, Nick, Kyong Nyon, Thomas Turin, Richard Bedrichki, Yada and Flo are no longer in the lab, but Flo, Kyong, and Nick are well known to some of you guys in Varna. Cody graduated 2 months ago. You know Cody as well. He was involved partially in the extracellular vesicle project. Cody graduated with 15 papers as co-author, and one in Nature Communications is going to appear very soon. So thank you very much. Nick, you can continue.
Thank you, Ilya. So as discussed, Nick will continue immediately, and the questions will remain for the end. So yeah, you have to stop to share and Nick to share instead his screen.
How does
Questions will be in the end, yes, Anton?
Yeah, questions at the end.
I will try to stay. Okay, good.
Please,
look,
yeah.
Anton? Anton, hello?
Yes,
Does the screen look good or?
yes, it looks good.
Okay, so on my presentation, I am going to try to concentrate more on the methodologies used for studying extracellular vesicles and some hurdles that we still have in understanding their biology and their significance in both disease and healthy brain. So Ilya talked about extracellular vesicles. Just wanted to reiterate that they are a heterogeneous group of lipid bilayer particles, and these are evolutionarily conserved. And then the term extracellular vesicle was first described in 1948 as platelet-derived particles, and it wasn't until 2005 that they were deemed important in the central nervous system. And the original studies were focusing on the role of extracellular vesicles in development of the brain. So each cell type is able to tune and secrete extracellular vesicles, and their excretion of this extracellular vesicles depends on their physiological state. And remember that extracellular vesicles are found in all biological fluids. This includes blood, CSF, interstitial fluid. If you're doing in vitro work in cell culture, they'll be secreted into the cell culture medium. And again, they're important in intercellular communication. Okay, so the extracellular vesicles regained interest in 2006, 2007 because it was noticed that RNAs were found, protected RNAs were found, including messenger RNAs and microRNAs were found in the extracellular vesicles, and that they could regulate the transcription of the recipient cells. So a lot of methods used to assess extracellular vesicles were not developed until the past about 5 years. And if we do look at a graph of the PubMed search of extracellular vesicles, you can see that starting in about 2010, you see this significant increase in publications related to extracellular vesicles. Right now, there's about 9,000 publications on PubMed related to extracellular vesicles. Of those related to brain, it's only about 900. Of those related to Alzheimer's disease, the number decreases to about 200. And an area of research that we're interested in is the function of microglia in Alzheimer's disease. And here you only have about 150 publications. Much of what we know about extracellular vesicles actually comes from cancer research, where you can see there's 3,000 publications of extracellular vesicles related to cancer. And Anton was talking about that earlier. There's quite a few challenges related to the field of extracellular vesicle biology. The first challenge would be that heterogeneous size distribution that we see. We see very different size particles that can be broadly classified, but it's not known if the size of the particles impacts function. So that's a challenge that there is. A second challenge is what is the contribution of the biogenesis pathway for the definition of extracellular vesicle biology? Again, can the size, number, and cargo of these extracellular vesicles be influenced by the cellular state? A third challenge is, can we identify redundancies within the extracellular vesicle cargo and the relationship among the extracellular vesicle cargo and the cell types? So addressing this issue may require development of elaborate single-cell vesicle analysis tools rather than the bulk analysis that is currently being used. Another challenge would be the target specificity. Are the nature and quantity of the cargo material sufficient to explain and ascribe biological effects? As a first approach, we could calculate more precisely how many bioactive molecule EVs, extracellular vesicles, can be delivered to the target cell. So that's one challenge. The fifth challenge we have is cell delivery. What are the mechanisms that underlie efficient uptake of the extracellular vesicles? Can we decipher mechanisms that underlie extracellular vesicle entry and the cargo processing within these cells? Can these extracellular vesicles merely bind to the cellular membrane and cause effect, or do they need to be internalized and cargo released? Sixth challenge is, how do the extracellular vesicles alter cell physiology? How is this cargo impacted? Once the extracellular vesicles enter the cell, do they need translocated to the nucleus to cause an effect? Can they be translocated to mitochondria, where they could potentially directly impact cell metabolism, generation of reactive oxygen species? And how do these changes in the cellular function impact on the function in health and disease? So do disease states associated with extracellular vesicles play a role in the disease, or is it merely a marker of the disease? That is a question that should be answered. And if extracellular vesicles are part of the disease progression or severity, can we therapeutically target extracellular vesicles to treat disease states? So I talk about 7 challenges, but there's actually a challenge 0 that should be considered when studying extracellular vesicles. Challenge 0 would be consideration for sample collection. I said that extracellular vesicles are located in all body fluids, including blood, plasma, CSF, but there should be considerations when collecting this, and it's important and it's the important first variable of analysis of extracellular vesicles. So when collecting, for example, for collection of blood, we need to make sure that the platelets are not activated and secreting extracellular vesicles, which could then increase the noise of the sample. So that's one consideration. And also when separating extracellular vesicles from postmortem tissue, it's critical to achieve a tissue disruption while minimizing cellular disruption. We don't want to destroy cells, which would impart intracellular components and artificial vesicle formation. We want to isolate extracellular vesicles through the interstitial fluid, not within the cells either. So if you have large disruption in the tissue, you may be releasing extracellular vesicles that would be normally found within cell types, and this could impact your analysis. So I was just going to quickly discuss two sample types that we're currently investigating in the lab. The first would be collection of blood. Whole blood by itself, it precludes extracellular vesicle analysis, so it really has limited application in the field of extracellular vesicles. Plasma is preferred over serum because with the plasma, you do not have the clotting factors that are secreted by platelet-derived extracellular vesicles, which again can impart noise in our sample. The advantage of blood is, again, it's an easily accessible tissue, and we can use it as biomarkers for neurodegenerative diseases. It's been shown that extracellular vesicles released from the brain enter into the plasma blood, and we can, again, use them for biomarkers in Alzheimer's disease. So there are some considerations when using blood as a sample for assessing extracellular vesicles. The first should be that the patients are fasted and collection is performed in the morning. You also want to choose an anticoagulant, which can affect downstream applications. This here is a table from the Izon group. Izon is a research company that does a lot of work with extracellular vesicles, and they provide some good methodologies on their website, so you can go there. But what we've been using is an anticoagulant EDTA, and this prevents clotting, so you don't have the release of the plasma or the platelet clotting factors, and it is suitable for RNA analysis. So in our human samples and in our mouse tissue, we've been collecting with EDTA. Also, again, you don't want to collect the first mil of blood. This can cause pressure and can cause contamination from fibroblasts. It's preferable not to measure extracellular vesicle function on hemolyzed samples. In our current study, we had a couple hemolyzed samples we included and found that they were not outliers in our set. So, you know, this should be a consideration when picking samples. And finally, you want to do steps of centrifugation, which can help you remove platelets, erythrocytes, and leukocytes. And you want to do this as soon as the blood is collected. That way that you have quality sample that can do extracellular vesicle analysis. A second consideration is when using brain tissue, if you're going to be using mouse models, it's best to do a perfusion of the mouse model with sterile PBS. We found that with the perfusion, you see a decrease in these cellular contaminants such as GM130 in the perfused mouse compared to the non-perfused mouse. Preliminary studies, you know, both with work we've been doing and other groups have shown that the post-mortem interval of the human brain-derived EVs really does not have a significant impact on the protein or RNA analysis of these samples. So while PMI may not have an impact, it still should be recorded and considered a factor when doing these analysis, especially looking at RNA profiles, proteomics. a protocol that we've been using in the lab is a very, it's not an aggressive tissue digestion that we're doing. You know, we don't want to disrupt the cells. We want to collect extracellular vesicles from the interstitial fluid without disrupting cells. So we use something very similar when doing single cell sequencing, where we incubate our tissue in collagenase type 3 and this hibernate solution. Hibernate solution is just a media that's helps support neuronal type cells. So we slice the tissue slightly, incubate it to dissociate. We then stop that reaction with some protein inhibitors and phosphostop, and then do some centrifugation and filtration to prep our samples for EV isolation. So it's a very simple protocol that we use. And again, you don't want to be overly aggressive with the tissue digestion. You don't want to be mincing the tissue or bouncing the tissue, because you want to minimize the cellular debris and cellular disruption. So the first challenge after having a quality sample would be the size diversity of these extracellular vesicles. And Elliot discussed that there's, you know, 3 main types: the exosomes, microvesicles, and apoptotic bodies, with these different sizes. Exosomes are about the size of viruses, microvesicles, protein aggregates or bacteria, and then apoptotic bodies are about the size of platelets. So there are a few techniques that you can use that are established techniques. There's also some novel techniques that are being developed. A good starting point would be this International Society of Extracellular Vesicles. They established minimal information for the study of extracellular vesicles. And that's one hurdle in the study of exosomes or extracellular vesicles, is that exosome research, it's plagued by inconsistency in the isolation methods. There's inconsistency in the nomenclature. There's a lack of standard data acquisition and analysis strategy. And that's what the International Society of Extracellular Vesicles, when they were established in 2011, was trying to standardize the methods and the classification of extracellular vesicles. So the 2 conventional methods that are supported by this society includes size exclusion chromatography and a differential ultracentrifugation techniques. There are also novel techniques such as immunoaffinity capture and polymer-based systems such as the ExoQuick system, and ultrafiltration systems that can be used to isolate different sized extracellular vesicles. Once you have isolated your extracellular vesicles, it's important that we then characterize the extracellular vesicles. We normally do this with Western blotting for cellular markers. Elliot talked about the transparent CD9, CD81, CD67, also proteins of the S-Kurt system, such as TSG101 and Alex here. So these are markers that you want to confirm are in your collected sample. You also want to include negative controls such as calnexin, which are endoplasmic reticulum markers, or GM130, which is a Golgi marker. You want to include negative controls showing that you don't have cellular contamination and you do have isolated exosomes or different extracellular vesicles. Once you've confirmed these markers, most groups then do a nanoparticle tracking, and this is just to determine the extracellular vesicle size. And you also want to follow that by electron microscopy so that you can look at the size of these extracellular vesicles and the shape. Electron microscopy is also helpful in identifying cellular fragments that are contaminants in your sample set. Again, Izon, a company I definitely suggest you look at their website. They've developed this tunable resistance pulse sensing, which is a single vesicle size determinant. Nanoparticle tracking system, here you're determining the particle of you're determining the size of multiple particles as they flow past the screen, where this tunable resistance pulse sensing allows you to determine individual sizes of single particles. So it's a very precise system that is far superior than nano tracking. So what have we been using in the lab? We've been using two techniques. We've been using the differential ultracentrifugation, and we've been using the Izon automated fraction collection. This is size exclusion chromatography technique. So these are the two classic techniques that we've been using. There's advantages and disadvantages to each technique. The differential ultracentrifugation technique can isolate relatively pure populations of exosome. And this is generally considered the gold standard technique for exosome isolation. This technique relies on separation of the particles based on size and density following centrifugation force and duration. The downside of the differential ultracentrifugation technique would be that it's very time consuming. For us to isolate exosomes from brain samples, this would take about 4 hours of our time, most of the time spent in the centrifuge, spinning down at different rates and at different RPMs. Also, there's an increased human error involved with the differential ultracentrifugation because you're removing the supernatant from what is a pellet of extracellular vesicles. When I say pellet, this is an invisible pellet that you find in your imagination, and you decide that is the pellet, and you need to remove supernatant from that. So you can decrease your yield based off of removing this supernatant. Recently, we've switched over to the Izon size exclusion technique. The advantage here is that we can perform isolations in under 30 minutes. So we can perform at a much quicker rate. Also, the cost is very minimal. The cost for differential ultracentrifugation is very expensive because you need high RPM centrifuges and the rotors for those centrifuges. So, there's a lot of upfront money to get that equipment, where with the size exclusion chromatography, it's just the cost of a column. One column costs about $50 and you can run 5 samples through it. So it's about $10 a sample. Also, there's hardly any human error involved here. And again, then after isolation, you can do the the nano tracking and confirm using electron microscopy. Here's some just articles showing the differences between. You can see on the left here, the number of particles using ultracentrifugation is very small, where using size exclusion chromatography, you're getting levels that are fold higher than ultracentrifugation. The size of the particles being isolated are not much different. And again, the yield using size exclusion chromatography is a lot higher than the ultracentrifugation. The only issue is that with the ultracentrifugation, you have minimal contaminants such as ApoA, ApoB here, where with size exclusion chromatography, you have increased contaminants from apolipoproteins. So it should be considered when analyzing your samples. This group then did look at a proteomic profile, and they saw really no difference between the two techniques. More recently, people have been using both techniques. And here's an example where we have the size exclusion chromatography. We have high levels of ApoA ApoB and ApoE compared to the ultracentrifugation technique. But when you combine size exclusion with ultracentrifugation, you pretty much reduce that level to 0. So you can, use multiple of these techniques to try to diminish the type of contaminants. But one thing to be considered is when you start reducing contaminants, you're also reducing the number of particles that you're actually isolating. So, it's quite a balancing act. And that's the same within the brain tissue. Here is looking at number of particles using size exclusion chromatography, followed by ultracentrifugation. You see high level of particles. They did some Western blots showing, high enrichment of the transparens. This is something also that we are doing in the lab. Ilya showed you the first figure from our publication. Well, that was with ultracentrifugation. We've now been doing with size exclusion chromatography, and you can see that the increase in the number of particles with the size exclusion chromatography compared to the ultracentrifugation. So again, it's just a choice on how you want to isolate the EVs, and what is your final analysis. If you're concerned with lipid numbers or lipids of this sample, then maybe you do want to eliminate apolipoproteins because they're transport a lot of lipids and cholesterol. But if you're more interested in protein content, then maybe it's not as important to remove those non-EV components. Challenge 2 was the biogenesis. Ilya went over the different techniques that the cell uses to release these extracellular vesicles. With apoptotics, you have the blebbing from apoptotic cells. Microvesicles is the budding, and exosomes is through invagination of the endosomal multivesicular bodies. One thing to note that EV production, it may increase in disease or damaged cells. Much of what we know about extracellular vesicles come from cancer. And exosome biogenesis is enhanced in cancer, with tumor cells creating more exosomes than non-tumor cells, and exosome levels in cancer patients are often elevated. But what do we know about biogenesis in brain? Some of interesting findings. Ilya talked about the SCART machinery and the Rab family, which is important. Also, GTPases and Rho. It's important in the rearrangement of actin, which is important in the microvesicle shedding. So those are two pathways, that we know in cancer cells. Also, reductions in sirtuins and disruption of lysosomal function. So once the exosomes are formed in the endosomal bodies, they are either trafficked to the membrane for release or they go to the lysosome for degradation. So if you have disruptions in lysosome function, you can see how that can impact the release of exosomes. And interestingly, this reduction in sirtuins is also seen in Alzheimer's disease. Alzheimer's disease is characterized by a loss of sirtuin expression, and you also see lysosomal impairment. So here you can see links on how Alzheimer's disease could potentially impact the biogenesis of extracellular vesicles. Also in cancer, we see that metabolic reprogramming of cells during the disease state actually impacts the release of extracellular vesicles. I included these studies in the presentation because our current work is focused on microglia, the innate immune cell in the brain and how it functions in Alzheimer's disease. And microglia, when they transition to different disease states, you actually see this changing and reprogramming of the metabolic activity of the cells. So this metabolic shift in microglia could potentially impact the release of microglial-derived extracellular vesicles. It could also impact the content of the extracellular vesicles, and these vesicles then could, send the signal to recipient cells, which could impact metabolic function of the recipient cells. So despite growth in interest in this topic, little was really known about this stimulation agents that can induce extracellular vesicle shedding in microglia. Most of the work is done in vitro, and that work shows that extracellular ATP through binding of the P2X7 receptor can actually increase shedding. Activation of serotonin receptors 2A, 2B, and 4, and also Wnt3 signaling can enhance the biogenesis of microglia EV shedding in culture. So there are some tools that you could use to help study the biogenesis of extracellular vesicles. Some of these tools include this compound GW4869, which is a neutral sphingomyelinase inhibitor, and this inhibits ceramide-mediated inward budding of the multivesicular vesicles. This is something that is independent of the SCART system that we discussed earlier. So here, using this compound, you can greatly diminish the number of extracellular vesicles that are being secreted in your model system. Recently, the group at Tufts University has developed an exosome reporter mice. So this mouse has CD63 linked to the GFP protein, and this mouse is under the control of AA virus. So you can increase, so you can have, for example, the expression of extracellular vesicles in particular cell types labeled with GFP. So in this first example, they labeled neuronal extracellular vesicles. They were also able to show astrocytic extracellular vesicles, microglia extracellular vesicles, and oligodendrocytes. Using this new model, they were able to show that the exosomes from neuron were not only transferred to astrocytes in vivo, but they also contained a microRNA 124. And this microRNA is important in the upregulation of glutamate 1 expression in the astrocytes. And this is important in the healthy brain for astrocytes to maintain a normal balance of the neurotransmitter glutamate. So this could be an invaluable tool to, do further research on understanding microglia extracellular vesicle release, especially in disease states. Extracellular vesicles, they do contain a rich cargo. Ilya discussed, these contain mitochondrial proteins, cytosolic, metabolic, and nuclear proteins, metabolic enzymes, metabolites, bioactive lipids, of course, the RNAs, non-coding RNAs. One thing to consider, these transmembrane proteins, these are important in that they can be used as markers to determine cell type origin and also cell type targeting. So studies have also showed that these EVs, they can be carriers of physiological as pathological markers. Ilya talked about, these extracellular vessels can contain tau and A beta, and these can be used as potential biomarkers in Alzheimer's disease. So what can we do with extracellular vesicles? You can, do many of the omics studies that are used normally. So some of these omic studies, we can do RNA sequencing of the genetic material in the extracellular vesicles. We can do metabolomics, looking at metabolites. We can do lipidomics, and you can also do proteomics. You can also do, PCR that we've done. We've done digital drop PCR for validation of our RNA sequencing results. Western blotting, looking for markers or looking for maybe cellular specific markers of extracellular vesicles. Some other things that we've done is we've done image stream flow cytometry. So this allows for imaging of extracellular vesicles. So you can label proteins, you could label RNAs, and look at the expression within the vesicle. Look if it's on the membrane, if it's intravesicular. There is also a kit called ExoGlow system, which allows you to label extracellular vesicles. So you can label the RNA, the proteins, or the membrane in extracellular vesicles. And then that way you can track their movement in culture or in mouse systems. So in the central nervous system, you have crosstalk between glia and neurons, and it's crucial for a variety of biological functions, ranging from brain development to maturation, homeostasis. So the central question is, can we determine what the barcode is that define extracellular vesicle source? What cell type are they coming from in the brain? And also, can we determine how they're directed to another cell type? And once they reach that cell type, what is the barcode on the target cell that responds to extracellular vesicles? So this is an important hurdle when studying extracellular vesicles. One thing to consider, especially when studying plasma extracellular vesicles, biomarkers, when considering plasma extracellular vesicle biomarkers, it's important to capture brain-derived extracellular vesicles. And then this can assist in monitoring of neurodegenerative diseases, changes, and it can help define specific signatures of the neurodegenerative disease. So there are some initial studies that suggest using L1-CAM, N-CAM, VAMP as markers of neuronal derived extracellular vesicles in plasma. So these could be used as markers that could help you identify extracellular vesicles coming from the brain that are found in plasma. Also, GLAST was considered as an astrocytic extracellular vesicle found in plasma. For microglia, there hasn't been much work trying to identify these markers. There has been one marker postulated, CD13, which could potentially be used as a marker to identify microglial-derived extracellular vesicles. Again, there is extreme crosstalk between extracellular vesicles from microglia and to microglia. Of importance or interest to us is the fact that extracellular vesicles can contain amyloid beta and tau, and these extracellular vesicles can activate the microglia. It's also thought that these extracellular vesicles, when phagocytized by the microglia, can then actually propagate the pathology seen, this hallmark of Alzheimer's disease. There's also extracellular vesicles that are released by neurons, and these vesicles are enhanced by potassium depolarization of the neurons. And when these extracellular vesicles go to the microglia, they help direct the microglia to remove degenerated neurites, and this is through upregulation of the complement proteins. There's also oligodendrocytes, which can secrete extracellular vesicles to microglia, and this regulates myelin removal and disposal of lipids from the oligodendrocytes. So microglia seem to play a central role in the communication through extracellular vesicles in the brain. So once the extracellular vesicles are secreted, the next step is to better understand when they reach the target cell, how do they deliver their information? How is the cargo processed? And this is normally seen through 3 steps. You first have targeting of the acceptor cells. You have entry of the extracellular vesicle. Again, this can either be entry into the endosome, where the cargo is then released in the cytosol, or it could be targeted to different subcellular organelles. It is thought that the membrane fusion of the extracellular vesicle, it is due to the pH found within the endosome. One area of interest to us is the blood-brain barrier and how extracellular vesicles can pass this barrier. The blood-brain barrier is composed of endothelial cells, and this prevents the unregulated leakage of substance from the brain. And there are 3 potential outcomes for extracellular vesicles at the level of the blood-brain barrier. The first would be that the extracellular vesicles bind and release the cargo into endothelial cells of the blood-brain barrier. This could also be of the endothelial cells of the blood CSF barrier and the choroid plexus. And once they release their content, this causes a signal propagation to cross the barrier. A second option would be that the extracellular vesicles bind to the endothelial cells, and these extracellular vesicles get degraded before passing the barrier. And this would help regulate the signal across the barrier. And then a third option would be direct trafficking of the extracellular vesicles across the barrier through some transcytosis mechanism. We and others have showed that exosomes can cross the blood-brain barrier. Using labeled exosomes, we've injected into the tail vein of mice and have seen the labeled exosomes in the brain. Other groups have also used radio-labeled exosomes and found that the exosomes can propagate in the brain. Also, exosomes that are delivered through the brain can be secreted out. Here is a study looking at radio-labeled exosomes in the release from the brain into the plasma. One thing we do know is aging is contagious. Everyone's getting old. So one of the things that we're interested in is learning more about healthy aging. And this is one study that we did where we looked at taking aged mice and treating aged mice with young serum that contained extracellular vesicles and young serum that was depleted of extracellular vesicles. And we can see an improvement in cognition of the aged mice with young serum containing the extracellular vesicles, where when we deplete the extracellular vesicles, they perform similar to the sham group. Other groups have done similar studies where this group here had injected old serum, extracellular vesicles from old serum into young mice. And this, again, was a peripheral administration. And we can see that there's increase in inflammatory markers in the brain following administration of these extracellular vesicles from aged serum. There is also the potential of the CNS-derived exosomes to signal to the peripheral immunity. And also, the peripheral immune system could potentially deliver substance to brain tissues. This second option is very important now, given the COVID pandemic and some of the neurological long-haul symptoms which are observed with infection of COVID. In this study here, a group treated mice with LPS and then isolated exosomes from plasma and then delivered those through tail vein injection. And they saw increases in inflammation in the brain due to these plasma exosomes, suggesting again that peripheral immune system can deliver exosomes and propagate inflammation to the CNS. It performed a similar study with high-fat diet, looking at how high-fat diet impacts brain inflammation through exosomes. And again, Ilya discussed the potential of exosomes as CNS biomarkers. Also of interest is the potential of environmental exposures to alter the release and cargo delivery of CNS and peripheral extracellular vesicles. One area of interest that we have is better understanding the choroid plexus and how the choroid plexus, which is the barrier of the blood CSF barrier, how it transmits its signal and potentially extracellular vesicles from the periphery into the brain. And there are studies just showing that that potential is there for extracellular vesicles to be transmitted. In the aged mice that were treated with young serum, we've collected choroid plexuses and plan to do studies looking at both choroid plexus function and potential microglia activity in those brains. There's also therapeutic strategies where you could load exosomes and take advantage of the fact that exosomes could potentially pass the blood-brain barrier. So you could load exosomes. Here is a study with exosomes loaded with catalase, and this improved outcomes in a Parkinson's disease model. And then also exosomes were loaded with curcumine, an antioxidant. And we can see that the exosomes, these exosomes were also labeled with a far infrared dye, and we can see that they were entered the brain, and they also had a better clinical outcome in this model of experimental autoimmune encephalitis. There's also studies looking at how microglia-derived extracellular vesicles respond to DBI and how there's a shift towards an anti-inflammatory and tissue repair expression profile due to the release of these vesicles later in the disease progression. One thing to consider is that microglia-derived extracellular vesicles differ depending on the priming status. LPS, which is a toll-like receptor 4 ligand, this increased levels of IL-1 beta, microRNA 155, TNF-alpha, and IL-6, while the same treatment with bacterial flagella failed to have that same effect. So there's this content-specific effect of microglia here. Also, LPS can stimulate microglia extracellular vesicles, and this can propagate the inflammatory glial activation. Another interesting study looked at how HIV impacts microglial extracellular vesicles and how when microglia are infected with HIV, it actually releases these extracellular vesicles, which can disrupt the permeability of the blood-brain barrier and helps increase the secretion of cytokines. It's also conceivable that there is some type of neuron-derived extracellular vesicles that can play a role in maintaining homeostasis in microglia phenotypes. So I've highlighted some of our work and others' work. I think it's interesting to see in the last decade the exponential growth of the knowledge within the field of extracellular vesicles. You know, we have a development of novel techniques, novel roles of extracellular vesicles in both healthy and pathological contexts. You know, there's definitely several studies that have contributed to increasing our understanding of neuron-glial communication. A lot of in vitro work to understand better microglia-derived extracellular vesicles, but again, there's still a lot of work and in vivo validation of these research results that are needed. I'd like to thank Ilya and everyone in the lab. And there's my email if you have questions on technical aspects of isolation of vessels, tissue preparation, feel free to reach out.
Yeah, thank you, Nick. First question: when you have a complex environment like in the blood, is there a way of finding out the source of exosome? I mean, whether it was derived from the brain, at least on this organ level. Then on the tissue level, if you have exosomes isolated from the cerebrospinal fluid, are they coming from the choroid plexus or from the ependyma? Or draining from, I don't know, subventricular zone or the cerebroventricular organs?
Is this a question?
Circumventricular organs?
Is this a question for me?
I think for Nick, technical question. Or?
Oh, it's a huge question.
Yeah, I mean, it's the biggest question, I would say. I think that's, you know, one of the hardest things, especially with using blood as biomarkers, is trying to figure out the tissue type it is being secreted. There are some membrane proteins that are thought to be neuronal specific. I talked about the L1-CAM and VAMP. GLAST for astrocytes, but again, these are speculations. You know, and then in different model systems, you do have the potential of labeling the exosomes and kind of seeing their transport, but not, you know, in clinical setting. Ilya, I'm sure, has more to add.
It's a very difficult question. It is one of the most difficult questions that people who work in the field of biomarkers are struggling with. So, and this is, by the way, one of the reasons extracellular vesicles in cancer are so, or the research of extracellular vesicles in cancer is so advanced, because you can not for sure, but to a great extent, you can monitor or you can identify extracellular vesicles in plasma or in peripheral biofluids coming directly from a cancer tissue. So it is it is possible, because you can obtain biopsy from a cancer tissue, and you can at the same time correlate the content or the cargo of the extracellular vesicles you isolate, which is practically impossible for brain. Now, the question for extracellular vesicles isolated from CSF is has the same answer. As you, Tony, mentioned, they may come from choroid plexus cells, they may come from ependymal, they may come from epithelial choroid plexus, and we don't know exactly, because we have seen extracellular vesicles transported through the barrier, blood-CSF barrier and blood-brain barrier. Now, what is the best way, particularly for brain physiology, development, and diseases, and not only, but for any of the extracellular vesicles, as far or when we are able to identify cargo molecules that are specifically or predominantly or exclusively expressed in brain or other tissues, then if we find those in plasma or CSF, then we can definitely use those either as diagnostic biomarkers or biomarkers that we can use to follow the progression or the result of a specific therapy. A year ago, a paper appeared published by a group. Tony knows the people. Andre Fischer was a senior author. Andre Fischer was in Varna in 2017. So they tried to associate extracellular vesicle cargo in CSF with Alzheimer's disease progression, and use those as diagnostic biomarkers. So what people do now is to combine or to assemble so-called signatures of non-coding RNAs in extracellular vesicles and associate those signatures with a specific disease. Now, why signatures? Because increasing the number of non-coding RNAs identified in extracellular vesicles, we increase the chance that they come from this or that, or specifically from brain tissue, because we know that they are expressed. But again, notice, they are also expressed in brain. So the signature that most of the people working in our field, in Alzheimer's disease biomarkers and extracellular vesicles, they know that there are non-coding RNAs in the extracellular vesicles that may come, and the chance that they come from brain are higher than from other parts of the body. So yes, this is a very, very, very important question, and it is a question that sooner or later will be answered, but not now. It is not answered now. We have some clues, and we will probably publish and disclose this very soon.
So yeah, I think that's where these omics studies are going to come into place, developing proteomic signatures, lipidomic signatures that would identify extracellular vesicles as coming from a particular cell type or coming from a particular cellular state. So without these, it's very hard to attribute an extracellular vesicle to a location or cell type.
Hello?
Other questions? Other participants?
I just can comment because I have always the same question which Anton raised, and I think that we need my name is Igor Reznik. Sorry, I'm now working together with Anton, but not in CNS, but in blood mainly, and immunity. And I think that for now, I'm absolutely agree with Ilya that for now we should make a negative answer about the source of things, about separation of things which we try to catch in the blood, for example, or in fluid somewhere with a specific tissue or tumor or whatever. And this is an extremely big problem, and I think that there are two different solutions of this problem. First one is that sometimes non-specific markers can work excellent and give some practically important results. For example, I don't know, fever, high temperature, of the body. It's has no any specificity, but it's helpful if, in many cases. For example, if you see pneumonia and you see high fever, you can associate and follow this fever as a result, as a sign of pneumonia and result of your treatment. If no, then you should make even differential diagnosis with. A more scientific approach, I think, is to make several models in parallel, not maybe in vitro model with target cells, with like we see in.
We lost the sound.
The connection of Igor, I think, is interrupted.
Yes.
Yeah, while waiting for him to re-enter, maybe anybody else questions? Diana.
Yeah. Thank you very much to both lecturers. I enjoyed very much the lectures. It talk me relatively new because I'm not working in the field, but several questions appeared when I listened to this to the talks. I was thinking about experiments with conditioned medium when we treat one types of cells with something and then we transfer the medium in we treat other types of cells. That means that we transfer also extracellular vesicles with a lot of other material inside, which has not been investigated. We only follow some markers, observe the results, but that's a very interesting thing. Then I was thinking about blood transfusion. Blood transfusion is also something that delivers a lot of genetic material along with, especially this convalescent plasma that is now used to treat COVID. And do you know if extracellular vesicles are used in some schemes for development of vaccines? Do you think that this is something that is in practice applied object of research? So yeah, many more things also were interesting to me, like the effects of curcuma, for example, that were mentioned. I haven't read about that, I have to admit that. So this is a huge new area to study, extracellular vesicles release as a response to different foods, for example, or physiological conditions. So really many ideas came out of this of these talks, and yeah, it's always nice to hear people, specialists in other fields. So maybe if you can comment something on that, I would be very grateful. Thank you again for your talks.
Okay, I will try to respond and address your questions, and then Nick will probably add some technical aspects. So conditioned media are very, very lovely object in extracellular vesicles research. The thing is that, yes, it is easy to obtain, and I agree that there are so many things within conditioned media. We have been working with conditioned media from transgenic and knock-in mice and all kind of AD model mice for years now, for decades. The only thing that I would suggest is, or I envision, is to isolate extracellular vesicles from your conditioned media and then instead of applying the entire conditioned media to other cells or to other animal models, use just the extracellular vesicles. Now then, you have to characterize the extracellular vesicles, which is not very difficult. People like us, they will tell you, oh, it's very easy. It is cheaper than what? If you want, and I strongly suggest, if you have experimental system with conditioned medium, you have to do whatever you have and can in order to continue with extracellular vesicles, because this is the cleanest system that you can find. Cheaper means in your lab, from the very beginning to the very end, of characterizing the cargo of extracellular vesicles, excluding proteomics and lipidomics. If you confine your research only to non-coding RNAs, you will have to assemble a set of equipment that approaches $350,000, almost $400,000. This is the cheapest. But it is doable. It is doable. Step by step, as we did, step by step, slowly, but very consistently, you will have this. Now, I will jump to the COVID-19 and extracellular vesicles. I don't know much about extracellular vesicles and vaccines, even though there are speculations on the web, particularly. However, there is a series of webinars that are organized by one of the most respected specialists and experts in extracellular vesicles. His name is Kenneth Witter, and the next seminar is on December 12th, and the title is Extracellular Vesicles and COVID. I will send you the website and this is I believe it is accessible to everybody in the world. The invitations go to the members of the Extracellular International Society for Extracellular Vesicles. But if I send you the invitation, you will be able to connect. Now it is outside our particular interests and work, but there are plenty of works about extracellular vesicles and nutrition. This is something that is very well presented in the field of extracellular research. But as I said, not because it is not interested, don't get me wrong, it is very much interested, but it is just too much for us. We try to be focused. We don't succeed, of course, because there are too many interesting things. And the better you have your laboratory equipped, the more the people in the lab go and they find all kinds of different things, and they think that it is doable. No, it's not doable because we don't have people, we don't have resources. So, Nick, if you have, so my understanding that there were three major questions, and I tried to address those. We can be helpful, particularly in handling conditioned media. We can send you protocols. You can do this. You can try this. It is not difficult, particularly if you have. And these are not very expensive. The size exclusion chromatography columns and collectors are cheap. You can really afford those. I know the situation in Bulgaria, but you can afford those. So you will be able to isolate and then to handle the extracellular vesicles downstream to your protocols. Okay, so Nick, now if you have something to add.
I would definitely say that you need to consider extracellular vesicles when assessing your conditioned media. And like Ilya was saying, these columns are fairly cheap. Technically, I don't even think you need the collector. You could probably do a manual collection. The collectors are automated. It's simply a scale and it weighs the amount of liquid that is coming through the column. So you could probably set this up on a precision scale if you have it and do a manual collection. And I would say the advantage of these extracellular vesicles is while their content is quite low compared to what might be secreted by cells, this is protected content. These are in lipid particles, protected from degradation. They can be transported long distances, so you can see distal effects from secreting cells. And plus, they are targeted. These vesicles have surface markers that target them to particular cell types. So, you could have lower content, but it being protected and targeted to a particular cell type, you could still see a large response when the signal is transferred. But I would say if you have any questions on how to isolate, don't be afraid to reach out. I would suggest starting with the columns. And again, you can do simple Western blotting or something like that to get some kind of initial characterization if there's some particular you're interested in.
Also, I would add that if you use these size exclusion chromatography columns, and if you measure at least once or twice the size and concentration of your vesicles, I'm not sure, but somebody at the university, medical university, may already have those instruments. So once you get confidence what you get at the tip of the column, then you can rely on this, because these size exclusion chromatography columns are very, very good, and the results are very consistent. If you do ultracentrifugation, differential ultracentrifugation, or gradient differential ultracentrifugation, you probably will get the same, but as Nick said, it's time consuming. And if you have everything, plasma or extracellular or conditioned media, it will take 2 days at least. There is no such thing that within 1 day you will be done. The columns are fast and reliable.
And that's also an advantage, because if you're assessing, cargo that could be easily degraded or something, with the columns, you have less chance of that happening than ultracentrifugation.
Thank you very much for the replies. Thank you.
You're very welcome.
Other questions?
Yes, I would. Can you hear me?
Hi, can I ask something? Oh, Dimo, please go ahead.
Okay, thanks. First, I want to ask if those vesicles implied in maintaining tissue homeostasis, for example, in stem cell niches, will promote or inhibit division depending on the situation around them. And the second question is, how stable are the exosomes and vesicles we were discussing here? Can we, for example, store them at minus 80 degrees and quantify them later for extended periods of time?
Yeah, I mean, these samples are pretty stable. That's what we do. We isolate. And normally, if we're going to assess RNA, we put them directly into Trizol for isolation and at a later point. But we do have vesicles that are stored at minus 80. A bit like with any sample, you want to avoid freeze thaw. Some of the techniques that are actually used to load these extracellular vesicles with drugs is a freeze thaw technique where you're kind of breaking the vesicles apart by freezing and thawing them and letting them form again. Related to homeostasis, there are extracellular vesicles released from neurons that help microglia prune synapses. So that's an important part of homeostasis. And we've also are in collaboration with a group that is looking at the effect of these extracellular vesicles on stem cell biology and the mitochondrial function, how it impacts mitochondrial function of stem cells and regeneration, both in brain and muscle tissue.
The only thing that I can add is that, as Nick said, we have extracellular vesicles stored at minus 80. The best way, if you are looking at RNA cargo, the best and easiest way is to store these in Trizol. So in Trizol, you can store those even at minus 20. However, the situation with or the requirements or the conditions, if your goal is protein content of extracellular vesicles, then it changes a little bit. But there are so well-established protocols that practically you can find a cheap and very reliable way to store those. Now, with the lipids, you may have already read and you may know that the lipids are one component of extracellular vesicle cargo that is less established, and there are probably not so many publications on lipidomics of extracellular vesicles, and the reason is that there are very subtle differences in lipid content between different extracellular vesicles. And you can imagine the reason is that these are membrane mostly the exact same extracellular vesicle will contain a lipid bilayer membrane that is specific for a given cell type or non-specific for any cell type. But there are methods, and we have some experience with lipids, particularly lipidomics of conditioned media. So you can reach out and we will give you all the protocols that we have. It's not a problem at all.
Okay, thanks both of you for the answers.
I think Trifon had question.
Can I ask? Thank you for the interesting presentations first. And my question is about, I don't know, several years ago, I don't know exactly 1 or 2, I found a very interesting publication for me, which was about peripheral immune challenge. It was a mouse model. So peripheral immune challenge with lipopolysaccharide, intraperitoneal challenge, resulted in pro-inflammatory state of microglia in the central nervous system. The interesting part was that actually the pattern of stimulation was important. So a single challenge, which peripheral challenge with LPS, resulted in pro-inflammatory state of microglia. And repeated challenges, like something like 4 challenges in given intervals, resulted in a tolerogenic state of microglia. This phenomenon has a very great potential impact on neurodegenerative diseases, probably Alzheimer's, schizophrenia, so on and so forth. So the authors didn't clarify the actual mechanism of this communication. So have you seen something about this, or there is a possibility after your talk that actually microvesicles may be the physical carrier of the signal from the periphery to microglia? Do you have some experience or something on that? Have you seen something?
Nick will probably answer.
Yeah. There are studies where mice have undergone that same LPS challenge, and they have isolated extracellular vesicles from the plasma and then administered those to naive mice. And you do see activation of microglia in the naive mice.
Okay.
An interesting thing about microglia, they seem to be one of the cells that uptake peripheral extracellular vesicles at the highest rate. So anytime you administer these in the brain, they almost preferentially go to microglia, potentially due to their phagocytic capacity. So that could be a potential mechanism of this effect. The repeated LPS challenge, I mean, that could be, due to a more chronic condition.
Yeah, probably it's just a reaction. I mean, that's a pattern which would suggest that this is not something infectious, so probably it's better to go down for the microglia. I mean,
There's models that we're using where we're looking at a more acute effect of amyloid beta to microglia, and we see a different signature than, say, a disease-associated microglia, a microglia that's been around an amyloid plaque for many months. You can imagine also that the metabolic changes in these mice or in these microglia are definitely going to be different. A microglia that's been around a plaque for some period of time is under high metabolic demands for long periods of time, where something that's a single exposure could have a different effect. And again, these extracellular vesicles with these changes in metabolic change, you could have increased release of extracellular vesicles that are trying to eliminate metabolic byproducts. And these vesicles, while the cell is trying to eliminate them, for that cell that might be good, but then it might be signaling to another cell that could cause some kind of change.
thanks.
What I can tell you in addition, very recently, actually a couple months ago, we have been involved in a study conducted by one of the principal investigators at the department, and I believe if there are people at the university, medical university, or at your department who are very much or who are interested in the effects of LPS and other things that I will tell you now, you can consider a seminar given by him and his people. So these people have been working for many years now on the effect of exposure to known exposure of mice to known carcinogens, in particularly carcinogens from the cigarette smoke. So the study was very well designed, very well designed. There were type of treatments with LPS, with the carcinogen, combined treatment, and also a compound that they believe has a protective effect. Now, I'm not going into details because this hasn't been published all yet, but I can tell you, I can tell you, and I'm not going into specifics, but I can tell you that most of the changes in the transcriptomics, bulk transcriptomics in brain of these mice was particularly, I'm not saying entirely, but particularly associated with LPS. And those were non-toxic doses of LPS. We know the effect of LPS in mice. You can kill a mouse. You can kill a mouse with LPS in hours. And they become disoriented, and you see the effect of the injected LPS within an hour. Of course, these are doses applied to kill them. But prolonged application of LPS in very, very, very small doses is something that is not very well understood. So the question here is, what is the practical usefulness of injection of LPS alone? So are we looking at the effect in brain or generalized inflammatory response or anti-inflammatory response? It is very difficult to say. But yes, if it is something delivered and transferred by extracellular vesicles released in the periphery, yes, it is possible. We know this, everybody knows this. These extracellular vesicles can traffic through the membranes. They can traffic transcellularly. It is also proven, particularly in the blood CSF barrier cellular content. So, but at the same time, you have immediate traffic of LPS to the brain. And there is a immediate response or prolonged response confined in the brain. So then the question is, where does or who does initiate the response in brain? Microglia, that particularly is under the attack of LPS, or microglia that is sitting there waiting for extracellular vesicles released by cells in the periphery, and then they go through the membranes and attack microglia. It's very, very difficult to discriminate.
Okay, thanks.
Other questions? Then if there are no questions further, I will
Are we supposed to see all this?
I will have to thank again the sponsor of this event, which I'm sharing. Do you see my browser now? I'm not sure.
Yeah, I see the screen.
Yeah, the screen, and I would like to thank our lecturers and consultants. And I'm sure in the next few days there will be some more questions. We have their emails here on the web page of the course. So when you click there, you know the website. You can always click to any of the lecturers on Friday and today here and write them questions in the following days. I would like also to announce one more workshop that is coming this Friday for in situ hybridizations, principle and practice, in collaboration with the Max Planck Institute in Göttingen, which you receive email with a link in the next few days. Ilya and Nick, do you want to say something else?
I just said, can you send me a link to that presentation?
I was just about to ask the same question.
Yeah, I'll send you. Also, we are going to stream it in YouTube here on this page when the time comes. But I'll send you also a link to participate directly.
Anton, thank you very much again for your nice organization. It was very interesting and useful. Thank you.
So,
Thank you, Yuri.
Tony, you saw the members of the so-called extracellular vesicles team in our lab, but there are other people, like 4 or 5, working on fish with RNA probes in brain sections. So that's the reason we are very much interested in this, and it will be very interesting for us to see the seminar if you send us the link.
Yeah, sure. Okay, so thanks everybody for participating, for lecturing, for sharing your experience, and yeah, we are looking forward to next meetings.
Okay, thank you.
Bye-bye.
Bye-bye.
You're very welcome, and everybody who has whatever kind of questions, just email us or call us. It is not a problem, and we will try to answer and help you if we can.
Sure.
And
Thank you very much
bottles
for
of
your
Rakia.
hospitality.
Bye.
Thank you, bye.
Thank you very much. Thank you.
Thank you, Diana, and have a nice day.
Bye. Thanks.
Thank you. It was good.
Hey, Igor, how are you? Yeah.
Switch. Okay.
The button on the right side.
Right. Yes, I see it. Okay. Okay.
Okay.
Nice to see you.
Everything's fine?
Listen, 80%, and it's very sad, but I cannot participate actively. Okay. Anyway, bye. We will be in touch.
Bye.
Bye-bye.